Hello? Oh, sorry. Hello, before we start the expert talk, I don't know if you received a notification, but I updated something on homework too. It's not about the homework itself, it's because I know some people were stuck with question 2, which is the hardest of the homework. So I uploaded a paper and the supplementary of that paper. Hint, please check the supplementary, it will help you a lot for question 2. So now you don't have to dig for it, it's just uploaded in the assignment. So good luck.
All right,
So let me make some announcement about today's expert talk. So the speaker will be Professor Ruby Huang, and she's in her way. But I want to talk a little bit about the, go to the next page, about the grading policy. So just to remind you again. Okay, so you need to submit your report of the expert talk on NTU Core, and that will count for 2% of the semester grade, and the deadline is midnight, November 12th. Okay, so please remember that, okay? So let's wait for a little bit. Professor Ruby Huang is on her way. Oh, and by the way, because some schedule change, we decided also some students are worried about having too little time to prepare for your paper survey, so we decided to postpone it. Okay, so next week we'll have workshop 3, okay, instead. Okay, and the TA will walk you through step by step. Okay, and we are still discussing with the expert talk 3 speaker to make sure that whether she can make it on the next week or the week after. Okay, so we'll make an announcement whether the paper survey will be next week or the week after next week. Okay, sounds good? You guys will be happy about this, so you have more time to prepare. Okay? All right, so next week will be workshop 3, and whether the week after that is the paper survey, we'll let you know soon. Okay?
你要充电是不是？它有一个拉，可以拉一下。可以拉。
老师，我帮你准备。这样会不会？这样没事，这样会拉过来。
主要是设备蛮齐全的。
好，这样，对，这够。好。这个是无线投影器，所以
哇，这么可爱，一直都
可以吗？
是今天
要电脑开了才知道。
就是有 response 的人就来。
哇，这么好，OK。看白有 response 就会加分。
哦，真的吗？然后但是因为是 trick or treat。
我要不是我女儿，我当然知道这个。
对，然后因为这个是在新加坡买的，20块钱。
有，刘老师登录在。
等一下。
我先关掉。
OK 了，对。
准备好了。
对。
那我就直接。
就是 teacher 给收的。
哦 ，teacher 给收的。
所以他是投到那边嘛，对不对？接到那边，然后对不对？
耶！
可以。
哎，等一下，然后这个有 HDMI，所以我如果要放影片，应该是可以。
可以，应该是投影机，会有声音。我调一下。老师要测试一下。
要测试一下声音。
它应该会有声音。
测一下影片，
测一下影片。
Since the discovery of DNA in 1953, the question of how it organizes itself
我今天就乱讲，
没关系啊，
就慢慢讲。
慢慢讲。你可以讲1个小时，然后后面会1个小时跟他们聊天，然后谈任何都可以，谈科学啊，谈生活。
真的吗？我本来打算慢慢讲，讲到2个小时。
你讲2个小时。
我不知道，
然后他们现在都有。
对，所以我其实有，所以其实我有一些 introduction。
OK， 那我就介绍你。
Okay, good afternoon everyone. So today we have the great honor to invite Professor Ruby Huang. She's a good friend of mine and a longtime collaborator. She's a professor of School of Medicine and also her expertise is on EMT, cancer genomics, spatialomics, tumor heterogeneity, precision oncology. And today we have the great honor she will come here and share with us gazing the epithelial mesenchymal plasticity spectrum in multi-omics dimensions. And she said that she also play trick or treat today, so you have a very exciting talk ahead of you. Okay, without further ado, let's give her a big hand. Thank you.
Thank you. This week is Halloween week, right? Are you guys going for any dress up, dress down? Don't know? Okay. All right. So I got this. You know what this creature is, right? Labubu, right? So I got this. This is a candy bag. So there are some treats here. I know this is like 2 o'clock, like in the afternoon. You probably are very exhausted. So for those who responded to me, you can come and grab something from this bag. All right. So I'm Ruby Huang. So I graduated from School of Medicine, trained as a medical doctor from National Taiwan University, and then I stay on to do a residency training in obstetric and gynecology. So I'm a real doctor. I see only women, but my husband, he sees men. He's a urologist. So if you guys have any medical related questions that you want to approach us, please rest assured he will not tell me what you tell him, and I will not tell him what you tell me, right? So the confidentiality. And then, so I finished my clinical training in National Taiwan University Hospital, and then I, just out of my mind that I decided to do a PhD. But if you are thinking of doing a PhD, I would tell you that it's a great idea. So, yeah, please don't hesitate. So, because I also need to finish my clinical fellowship in the hospital to specialize in oncology. So that really limits my choice of going abroad to do a PhD. So I decided to stay on again. in NTU to do the PhD study in the Graduate Institute of Anatomy and Cell Biology. So you will see that I was told that today we are going to talk about a lot of different things, but I will mix everything into the lecture. So you will see that sometimes people will give you advice that don't stay in the same university for too long. Reason being that you will be mixed with a whole bunch of the same students, the same faculties that will limit your kind of exposure. But for medical doctors, in practicality, if we want to take care of both medicine and then the advanced study is sometimes not easy to break away from where you are. But for you guys, I think the sky's the limit, right? You have a lot of choices where to go. Of course, NTU is a great university, so if you still have strong bond to NTU, we will of course welcome you to stay on, right? But you're also encouraged to venture outside. And then, unlike many clinicians, I decided to do further go into research. So I decided to do a postdoc. So if you are in biomedical field, unfortunately, postdoc, at least one postdoc, is the minimum to get you anywhere, even in industry. All right, the industry starting job is actually PhD in biomedical. If you're only having master, you will be doing those like routine jobs, which could be easily replaced by even no. Let's don't go there. All right. So, but at least you need at least one good postdoc. All right. I'm not sure whether for computer science is the same thing. No need, right? But for biomed, at least you need to have one postdoc. The choice of postdoc. I did my postdoc in Singapore in a research institute, but now in biomed for postdoc you could actually go to industry. There are many pharmaceutical industry that they also offer good postdoc positions, right? Even I think Google, OpenAI, they also have postdoc positions, right? So and then after that you can decide where to go. You can either stay in the industry. If for medical doctor, for us the industry is hospital, right? But for me, I can decide to stay on in the research field. So I continue in the research field. So I stay on in the research institute. Now come back to become a faculty. But you can see that my interest is actually quite broad. My affiliation is now with College of Medicine, but I'm also affiliated with engineering, and then also the International College, and then also D school in the design school in NTU. So my interest is really multi-dimension. So how do you develop a multi-dimension interest? I think it's because I'm really kind of even I'm introvert. I'm actually an introvert, but I don't appear like an introvert. So people come to me, and then we will strike interesting conversations. So that's how I will interact with people like Professor Zheling, computer science. I will interact. I have a lot of friends in sociology, social science. Like my husband, he ventured into public policy and the public health, right? So through this, you kind of broaden up your dimension in your career. So don't if you just start from I don't know in this computer science and then heavy course, but I presume a lot of you guys come from different discipline, different backgrounds. Right? So engage with people, strike random conversations. I just recently attended a workshop called Serendipity, and then it's held by a professor from University of Tokyo in Department of Chemistry. He believes that science, all sorts of science, would generate from serendipity, just from random chatting. You know, sometimes you caught someone in the elevator. For example, yesterday I was in the lift with a young faculty, and then he say hi to me. I say hi to him, and then we started chatting, and then we just like, you know, just suddenly he said, "Oh, I'm working on RNA binding protein." I said, "Oh, we also fish out one RNA binding protein," and they said, "Oh, what's that?" And then we started to chat, and then like 30 minutes later, we have a project already, all right? So just like that. So, you know, be open-minded, chat with your friends, even though it might seem like very random, all right? It's too long of an introduction, but this is what I do in the lab. So I have 3 main interests. One is called the epithelial mesenchymal plasticity. I will spend a lot of time going through this with you today. And the second thing is the ovarian cancer research. Like I said, I was trained as a OBGYN. So my subspecialty is GYN cancer, particularly is ovarian cancer. So I spent some time to talk about why we are doing this in Taiwan. And the third interest is I have this period education. I don't know whether some of you might have, no, probably not possible. Because, so this is a general education course that it is for undergraduate. It talks about period. Is menstruation. So it's quite popular. It's Asia's first general education course, and then it's now the only general education course in the world. You will be surprised that both men and women will sign up in this. Now we almost have half and half of both men and women in this class. Actually, we are every Wednesday. From 4:30 to 6:20 is our class slot. So, you know, after this, I go into the periods at the Boya. If you're interested in what we do, you're welcome to join, talk to the students there. All right, come back to science. So I will introduce something called epithelial mesenchymal plasticity. So the name suggests that there is some change. Plasticity means change, transformation between 2 states. One state is called the epithelial state, and the second state is called the mesenchymal state. Who can tell me what an epithelial state is? Any one of you have taken my course on anatomy and physiology? No? Some of you? What is an epithelial state? These are TAs, right? Oh, so they're not supposed to answer. So these, okay. What's epithelial state? Okay, what is epithelial cell? What is an epithelial cell? So epithelial, no idea? No idea? Epithelial cell is the building block of your body. Basically, your skin, your gut, your hair follicle, they are all lined by epithelial cells. Okay, and epithelial cells, they are kind of like held on to each other. Like for example, this group, right? 4 of you, each one of you can represent a cell. And then when 4 of you are like tightly packed together, it's kind of like how you assemble this table, right? You become like a line of cells. And then imagine you have multiple lines of cells lining up together. It becomes a tissue. So from a cell, you become the tissue. And from tissue, you will the different types of tissue will fold into different types of organs, right? For your skin, it's a sheet, and for your gut, for a stomach, it's like a bag. Okay, so epithelial cell is the fundamental building block of our body. Of any living organism. Without the epithelial cells, there won't be tissue being made, there won't be organs, so you won't have a living organism, all right? So this is essentially the basic building block. And then for epithelial cells, like I explained to you, the cells need to be held tight with each other. It's kind of holding hands, all right? And then like in between each epithelial cell, you will have the glue. Okay, these glues are made by proteins. These proteins are called junctional proteins. You will have this tight junction, you have this adherence junction, you have this desmos. So don't worry about this jargon. So you will have different types of proteins making up different types of junctions. And then you will have this actin belt. This is called the cytoskeleton, which is kind of like, you know, like a cable. And then traversing through the epithelial cell to holding it, to keep the shape, to keep the epithelial cell as like a shape. It's kind of like this bag, right? You have these strings here. So it's like keep it in shape, right? Imagine you have many Labubu black bags lining up one by one and become a Labubu tissue, all right? Something like that, all right? And then, so this is epithelial cell. And what about mesenchymal state? What about mesenchymal cell? Anyone knows what a mesenchymal cell is? Do you know fat cells? Fat cells are a type of mesenchymal cells, like a bone, cartilage. Those are a type of mesenchymal cells. So anything that is not epithelial cell, that's mesenchymal cell. Mesenchymal cell is kind of supporting tissue inside your epithelial tissue. So a normal tissue will consist of these two different states. So when we say that when there is an epithelial mesenchymal plasticity meaning, like I said, meaning a shift, a change, meaning that you will change from one state to another. So this epithelial state changed to the mesenchymal state, right? So it's called epithelial mesenchymal transition. Is it possible? Is it possible? You said, why is it possible? When do you need to have this plasticity? Can you think of one? When do we need to have this plasticity? Okay, like you have a wound in your skin, right? You have a cut in your of your skin, and then you damage the epithelial cell. Some of the epithelial cell will be gone because of the cut. And how would the epithelial cell, how would your skin cell to repair the wound? How? So what happens is the skin cell, the epithelial cell, they will need to double, they will need to proliferate, they need to make 2 of them or 4 of them. They need to proliferate. And then when they proliferate, they cannot just, let's say this is the wound, they cannot just all like clutter here, right? They need to go and cover the wound there. So when the cells need to go there and cover the wound, meaning that they need to leave where they are and then migrate to where that wound is. So wound healing, when you have a wounding, you have a wound healing, this is where physiologically you will have this plasticity. And then it is reversible. This is reversibility. It's called mesenchymal epithelial transition, MET. So it is reversed. So it's kind of from mesenchymal state back to epithelial state. Is it possible? Is it possible? You keep nodding, but you never say anything. Is it possible? Yeah, give me one example.
Just after the migration, then the mesenchymal cell will try to transform to the epithelium cell.
Perfect, exactly. So after the wounding process, right, your epithelial cell migrate to that wound edge, lodge there, and then the wounding epithelium needs to reform to cover the wound, right? So meaning that all the epithelial cell needs to hold hands together with each other. So this is where you would need to have this reversal of mesenchymal epithelial transition. So it's MET. So it's reversible, right? But the name would suggest that it's only switched between these 2 states. So you will only have 2 states, either epithelial state or mesenchymal state. It only shifts with these 2 states. But in fact, it is not. Because you know that when you need to break up with someone, It's never 0 to 1. It's never black and white. You will go through painful process of breaking up, right? Those painful process is sequential, or if you want to engage a relationship with someone, it's never like just like that. Well, sometimes it happens, but usually, normally, you will go through a process. So you will have a lot of transition in between. So it is never a binary process. So for this plasticity, for this cellular plasticity to happen, you need to go through many different steps. So we call this like intermediate steps. So in fact, this is an entire spectrum of that cellular plasticity. From one end is a very epithelial end, and my pointer, and the other end is a very mesenchymal end. And then in between, you will have this mixture of states. You will have this early, more E, a little bit of M. You have this here is like you will have equal E and M, and towards the M side of the spectrum, you will gain more M. And then finally, you will switch off entirely the E, and then to have the M. You got it? So it's the entire spectrum. So you will think of it like, is it a good example to say transgender? No, I probably don't want to go there. Okay, it's like personality, right? When you want to acquire a certain personality, let's say, okay, have you all heard about MTBI, the 16th personality? You will be tested as 1, right? Like I said, I'm introvert. But if I want to become extrovert, switch. Sometimes it's very hard to switch. So I will need to abandon a little bit of my introvertness first, gain a little bit of extrovert, right? And I might be or might not be possible to be fully extroverted. I might be stuck somewhere in between. All right, so this is kind of similar plasticity. All right, and you will see that at this entire spectrum, you will have this called the metastable and the stable state. What does it mean by metastable and stable state? Anyone from physics department with physics background? No? Yes? Okay, so metastable state is a physical term, is a physical phenomenon, is a concept in physics. So metastable is in contrast to stable. Stable state usually is where you have the least energy. And metastable is where the energy level is up, but it's at a delicate equilibrium. It is still in equilibrium, but it's not stable. So with a slight perturbation, this equilibrium will be changed, and the energy will be released, and then that state will be changed to a stable state. So this is called a metastable state. So whenever you need to have a plasticity, this applies to anything. This is cellular plasticity. You can have it for tissue re-engineering, whatever. Whenever there's a plasticity, you need the cells, you need the state to become a metastable state before you really switch into the stable state. And then one example for this is supercool water. Anyone know what is this? Supercool water? Have you watched it? Let's. No, it's okay. Let's watch supercool water. Mm-hmm.
想要6折的牛肉面，省省吧，叫富喷达送，输入优惠码天。
chill like that, I'm 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 chill.
So one day, this high school experiment, right? They put a bottle of Fiji water. Suppose it has to be Fiji because it's the purest, right? So you put the bottle, and they're expensive too, and you put the Fiji water inside the fridge, inside the freezer, before it actually become like rock ice. So the temperature is already below zero, right? Supposed to become ice. So the water is a liquid state. The ice is solid state, right? But before it can crystallize, you take the ice bottle out. It is still water. It is still in liquid state. But the temperature is already dropped inside, right? So the H2O molecule already has been kind of primed to be crystalline, to transform in that state. And what happened here is they shake it, perturb it, so you will have a seed where it could crystallize, right? Or you just put it out onto another cold surface, right? The other experiment they did was just to pour it out on the normal room temperature. It doesn't work, right? So we pour it out onto the cold temperature, cool surface, and then it will, kind of like frozen, like a slushy type, right? And it will change the state. So this is when that ice bottle water is in a metastable state. And then that perturbation, either you shake it or you pour it out, that's the break of equilibrium. And then the change of the state into ice or into the slushy, that is make it into a stable state. All right? So the concept of metastable, when you think of it into when the cells need to change the state from epithelial state to mesenchymal state, it also need to go through this metastability. And then to overcome this, there will be barriers. So the energy barrier. In this case, this is a thermodynamic law, right? So this is from water to ice. But in cells, you will overcome different barriers when the cells need to acquire the plasticity. Okay, so this is where we need to think of how, in the field of computation, because those can be put into numbers. How do you quantify those plasticity? How do you measure those barriers? All right. So, like I said, it's never just binary. It is a spectrum. You will have cells that need to, from the very, this is very epithelial state. You have a very tall cell with apical and basal polarity. The cells are very standing up very nicely, and then the cell need to slowly crumple down. So it's kind of like the Labubu bag is kind of going down, and then eventually the cell going flat. So you think of how when the cell needs to go through each step. First of all, the gluing between the cell need to be disintegrated, meaning that the junction between the cells, the protein, needs to be disintegrated. And then the belt, the actin belt that hold the shape of the cells, needs to be loosened up or eventually be broken down. And then the cells will need to change the shape from a very tall cell to a very flat cells. And then the cells usually, initially, they don't move that much, and then we start to crawl to gain motion. So each step, you will need to overcome certain barrier, okay? So to establish that entire plasticity spectrum. And inside this epithelial plasticity spectrum, it is actually controlled by different layer, DNA, RNA, and protein, all right? So why am I interested in this? Because during cancer progression, this is actually happening. So it's not our imagination. So you think this is a normal epithelium. Think of it as your skin cell again. You have a normal skin tissue, even though the skin is multi-layer, but never mind. Just think of it as epithelial cell. You have this normal epithelial cell sitting nicely with each other, and then forming a sheet, right? And then somehow you will have this early event of dysplasia. This is an early tumor adenoma, dysplasia, like polyp. You guys know about polyp? You have like a growth, but that's benign growth. That is not malignant growth. So when you have that, the cells need to lose that architecture. They need to become not the polarity of the cells needs to change. And then you see the cells start to sit on top of each other. They are not sitting nicely on the chair holding hands, but they start to be like randomly sitting on top of each other. And when this normal but like overgrowth of cells continue to undergo carcinogenesis, this is called carcinoma in situ, meaning that they are kind of still stage 0 cancer. You can see that this array of these cells become more severe. Not only they sit on top of each other, they kind of like start to organize in a more disorganized manner. And then further, this is called a stage 0 disease. And when carcinoma started to spread out, to invade, to become stage 1, stage 2, stage 3, stage 4 diseases, one thing they will do is the cancer cell, like I said, they will start to gain the ability to move out of where they are. It's kind of, some of you guys like come to Taiwan, right? You could stay in your home country within your home, your comfort zone, but you break out, right? You travel abroad to Taiwan to study. All right, so you are those leading cells, okay? So those are the metastasis cancer cells. They also do the same. They need to gain motility. They need to adopt new identity. They need to acquire new ability. Probably you need to start to learn Mandarin. I'm not sure, right? After you land in Taiwan, right? So nevertheless, these epithelial cells, they need to change some identity. They need to abandon some their original epithelialness and acquire the mesenchymal state. And then we all know that those metastasis cancer cells, they will go through circulation to metastasize, go into the circulation. Have you heard of something called circulating tumor cells? Those are the cells will go inside the bloodstream. And then those cells, those single cells inside the bloodstream, they are very vulnerable to be honest. Imagine you alone being injected in, floating in the wilderness of North Pole, right? Without any support. It's very easy to die off. So these cells inside the bloodstream, they need to acquire some resistance to die. They need to acquire the resistance to die. They live, and then amazingly, they venture out of the vessel, go out of the vessel, reach to the final destination, maybe liver, maybe lung, maybe brain, anywhere, to establish another colony. This is called the metastasis, right? And then, just like what we say, when they reach to this new destination, they need to reform that epithelial colony so that plasticity is reversed again. Yes?
Maybe a naive question, but how do they all end up in the same destination? Why
They don't all end up in the same destination. It's a good question. Where do they go? You would have, how do they know? Yes, exactly. Let's say, like you from Poland. No, you're not. Oh yeah, sorry. Like, okay, never mind. You decide to go out of your home country, right? A lot of your friends, they make the same decision, but they will have a choice of where to go. So how do you decide? Which place attracts you more, or where you have the opportunity? This is the sea and soil theory. So the destination you will have a cue. Say, for example, liver will have a cue towards a certain population cancer cell. Say, hey, 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 come here. And you will have the receptor and the ligand pairing from that cell to the destination. So that will attract it. This is one, right? And the second possibility would be, it would just be random. I would just go anywhere and then see where has the best soil to keep me there, right? So you will traverse to first destination, whether that's favorable. If it's favorable, I will stay. If it's not, Taiwan is too crappy, I want to move again, to the next destination. So there are several different ways for them. They don't all end up in the same place. Yeah, but nevertheless, they all need to go through this plasticity to be able to move out from doing this progression. So this entire phenomenon was discovered by my postdoc boss, Professor Jean-Paul Thierry. So back in 2002, so it's already 2 decades old. So if you look at this, is exactly inside the schematic representation of what is happening during cancer progression. Actually, inside the tumor cell, we know that the tumor cell is a mixture of different cells of different identity. So you would have cells inside the tumor already in epithelial state, in a mixture of the different epithelial mesenchymal state inside it. And then when metastasis occur, they don't travel alone. They travel with their friends. They metastasize into cluster. They engage into the communication with other cells, for example, immune cells or fibroblasts, the mesenchymal cell. And then somehow, like I said, they survive and then re-establish their colony at the metastasis. So the entire process, the cancer cell need to maintain their identity at some point. They need to shed off their identity. They need to acquire new identity. They need to be very plastic so that they will survive, benefit. Because for cancer cell, the only way, that they need to gain the growth advantage, right? That's their end goal, right? Question?
So if you go back to one slide before, what is the time frame we're talking about?
Is actually very long. Okay, for example, from the normal epithelium to the dysplasia, it could occur like a decade. Because for example, how old are you now? 29. 29, right? So it's actually very rare for a 29-year-old to have abnormal growth, for example, from your colon or from anywhere, because you guys are young. The cells would have a very good repair mechanism. Even though you smoke, even you go into the sunlight, you have the carcinogen, but your repair mechanism is still good compared to me. I'm 51. My repair mechanism is much worse, right? So I will have, if I exposed to the same carcinogen, I probably were more likely to accumulate those damages, right? So let's say if you continue to smoke, let's say from 29 until like 39, so normally, when will you start to get the first hint that you might get a lung cancer? Now it's getting younger and younger, but usually 29 is not the age you would hear of people getting cancer, right? So this is a very, very long process for normal epithelium to acquire those insults from your behavior, your diet, your environment, and to go on to this entire process. It takes decades. But you all hear about like you will have fast-growing tumor, right? Fast, advancing cancers. So it will accelerate. That means that the barrier for the cells to fend off the normal equilibrium will be much faster. So when the cells, once the cells start to become invasive, invasive, then it goes to stage 1, stage 2, stage 3, stage 4. It could be very fast, much faster. So it's not an equal time frame we're talking about here. So the plasticity, so that comes back to the question, when we talk about the plasticity, what about the time sequence? Is there a time? Because we probably will assume that from first shed of first feature to the gain of the last feature, you will go through a linear time progression. In fact, it's not. All right? So if you want to build any modeling for that related to the time, then the time is not a constant linear projection. It's a very good question. All right. So I spent a lot of time explaining this concept. It is just to lead you to this, because this is a very important concept of cancer biology. It's called the hallmarks of cancer. All these functions, and each one of them, is describing one way for cancer cell to survive, to kill their host, to establish their colony, because that's what cancer cell wants to achieve, right? All right. But unlocking the phenotypic plasticity allow the cells to change when they need to. Let's say if you want to kill the cancer cell with treatment, you treat it with chemotherapy. Some cancer cells, if they can acquire the plasticity to change it into a form that the treatment cannot kill them, right? Then the cancer cell will survive. And then, so it's kind of like a great, put on the great show, like masquerade for cancer cell to adopt during this entire progression. But cancer cells, they don't do this, they don't invent things. Most of the time, cancer cells, they don't have any innovation on their own. They adopt things that the body are already using, right? So usually the cancer cell will adopt a mechanism from developmental program, meaning that from when you are still an embryo growing up into adult human being, right? So EMT is actually, the epithelial mesenchymal plasticity is actually this kind of mechanism that is not only important during development, but is also evolutionary conserved. When I say evolutionary conserved, it means that you can identify the same mechanism in a lot of different species. Here I'm giving you 3 examples. One is cnidarians. These are the non-bilateral, okay? And Drosophila, this is a fruit fly, and sea urchin is the uni that you will eat if you go to Japanese sushi, right? And during these 3 species, these are our animal kingdom. They are at the very, very bottom of the kingdom. It's very far away from us as Homo sapien, from the mammalian. So you can see that. Don't worry about this mechanism. You can see that you always can find EMT happening during their very early stage of embryogenesis, during very early stage of when a cell of certain epithelial state needs to become another state. They need to undergo this EMT to acquire that mesenchymal transition. Okay, so again, tumor cells, cancer cells, they don't invent, they just acquire whatever has to be invented throughout evolution, throughout development. All right. So this EM plasticity, it will contribute to the heterogeneity of the tumor cells. Because the epithelial state, mesenchymal state, they're just the state, right? I know I shouldn't go for it, but let's just give it like, gender. It's binary. But imagine you could have a spectrum of gender, you shedding one and gaining the other. Okay, I'm sorry if you feel this offended, but that's, you can think of it that way, if that helps you to understand it more easily. So the epithelial state, these are just a state, right? It's kind of like binary state, epithelial state, mesenchymal state. but like for gender, you underneath that gender spectrum, if you want, you would still have a lot of different personalities, right? You would still have extrovert, introvert, okay, right? And then you will have other attributes for that, right? So it's the same as tumor heterogeneity and plasticity. So with along this entire spectrum, each individual cells, even though they might be at the same state of that plasticity, that same mixture state, but they would still be different, you know? They would still have different DNA mutation, they would still have different RNA expression, they would still have different protein expression. So what we are looking at is the phenotype. It's what it appears to be, the phenotype. But underneath that phenotype, you would have genotypes, you would have the transcriptomic state, you would have the protein state that still gives rise to the heterogeneity, the diversity there. And then all together, that constitute into a diverse ecosystem there. Am I making myself clear here? Yes? All right. So I won't have time to go through all this, but you would know that there are people are trying to target this EM strategy, EM plasticity to kill cancer. These are the strategies to target the plasticity. You can either kill cells at different state, or you can either push cell to one state, or you can, convert the cells, into a mixture of state. So there are different ways to engineer this. And why is it important? Because when a cancer patient is getting treatment for their cancers, the cells that are being killed versus the cells that are left behind, they might pose different EM state. So at some stages, some cells with more epithelial state are easier to be killed. Some cells are more mesenchymal state, they are easier to dodge the treatment. All right. So that's why it is important that we understand how the cells acquire certain state, how cells overcome certain barriers to go into the next state during the entire carcinogenesis progression, also during the entire treatment course of the patient. Okay, I think I will stop here. So that's it.
Yeah, we have question, comments.
Later on?
Yeah, no. I think you should share the Labubu with the student who interact with you. Okay, so student who interact with Professor Wei Huang, please come forward and get some candy from the Labubu bag. Yeah, come. Don't be shy. Yeah, come. Yeah. Yes. Anyone who spoke in the, yeah.
You're just ready. Actually, I have enough work. You want to change it?
Thank you. Okay, we still have a lot more as I can pick from the back. So please, student, as this is a rare chance you can interact with Professor Wei Huang, who's a really important figure in the cancer community, especially for ovarian cancer and also EMT. So if you have any question, we'll reserve the next hour for your question. But before that, let's take a 10-minute break. And when we come back, we'll continue our discussion. Okay? All right, so see you after 10.
对，因为我就是在下面，我就是要讲说怎么去 compute 这件事情。
对，你可以还是花一点时间讲，因为花他们会觉得好像。
为什么要讲这些？
对，只是简介而已。等下你回来稍微看。
我就是
就是下一段就是要讲怎么样考虑
算那个 score，
对，怎么算这个 score，因为后面
Marta 他来太早，
对对对。
那你可能再花15分钟讲一下，然后我们再跟
我应该就会停在这里，因为后面就是讲到自己那个不讲到应该就是讲这个怎么算。
OK， 好，感谢。
真的不要吗？
会的，等一下会。
Because the cell structure is different.
It's already Sorry about that. Ask me about that.
Because you need to upload it from the system.
Yeah, I think so. That's fine.
If you go for 5
Yeah, but I'm sure you get it. Don't worry.
Yeah, that's how it works. Like, in my lab, they already make but it's not that they are not able to provide some different methods. Like instead of microbiome, the biology
Thank you.
I think so in future it is going to be very common.
但是因为我觉得我如果没有把这个 concept 讲清楚，
Because right now the life
对，后面他们才说那个 computation 要怎么去做，因为就好像是我们曾经说 phenotype 定义的 phenotype， 它里面会
like that. Like right now we are more towards the not the sugar, it's completely bad.
所以你的 phenotype 是最上层。
对啊，其实下面还有很多很复杂的造成它的结果。
对，所以你如果要去 model 去算这件事情，你要去算它的 phenotype， 你就变成说你要算的是什么，你要对应的是什么，对不对？你懂我意思吗？
你说对应的是，只是说，我以为你EMP score 是从系统态的一个
对啊，是从 gene expression 开始算，可是那个 concept 就是说，它算的是最后 RNA 遇到，可是它的 DNA 是不一样。
对，没错。
因为我其实后面就要，因为讲到 DNA 就更难，就要讲到
Like in my lab, our
那个怎么去 model， 因为那个 DNA 的 accessibility 你其实又会影响到
if you are not able to
transcript， 虽然我们现在都把 transcript 当成是
how you are going to treat.
对，
但是从 transcript 到 protein 又是
对啊，又是一大步，
因为 protein 还有所谓的所以就是我觉得可能跟
Marta is dying to talk to you.
I have actually questions to both of you, but they're slightly unrelated. First of all, I wanted to thank you for helping me with the emails. Because I asked Professor Ruby because I want to do like PhD work more related to what she does. And then I texted her about advice, and she gave me advice. So thank you. Yes. Do you think like the West is slightly better? Like in terms of like the availability of labs? That's just like
while in the States. But I just don't know how are they accommodating like Harvard slashing people.
Yeah, it's a bit crazy. Yeah. I've been hearing.
But I want to go into like there are different types of cancers. All cancers happen in the same way or
Yeah, that's why I'm here.
so why is breast cancer is more common as compared to
I think Australia sounds great, but I think I would get disowned by my family because it's like the other side of the world. I think it's a bit too far for them.
Oh, I thought that you have some conflict with Australia.
No.
I was a little bit shocked.
No. It's just it's like exactly on the opposite. It is an option. I just, to be very honest, I hate the weather.
That's something we cannot fix.
That's the thing. There's nothing we can.
Oh, but now you can have co-supervising with international supervisor.
Well, I thought because I was sitting at the but yeah, I want to thank you because I wasn't expecting such a nice conversation.
Yeah, because I think you're really interested.
Maybe not ovarian fertility, but in general, gynecology.
Even that is nothing. I think UK is.
Yeah, there's a big institute in Oxford.
And I have like
what do you guys think about it?
but if you see the weather is
going tomorrow.
I already lived
The UK
there.
weather is not
So
really.
I have to say it's much different.
there must
I've
be.
been like I've lived in the UK for 3 years, so if you want from something, and I have to say I think I prefer the UK weather.
Because it's too hot here?
It's too humid.
Too humid. Oh.
In UK it's dark and it rains, but it's dry. But you know what? If weather is the only thing that will be keeping me from a PhD, then I think I should relocate. So I'm not going to be actually.
Yeah, that is true. That is
I
true.
will not be thinking about the weather as like the final.
Oh, that is on me. The pressure is on me.
No. She's just saying that weather is the biggest factor, then probably she's not so into PhD.
So I will not actually. I mean, I will be thinking about PhD, but I will be evaluating it based on this.
Yeah.
That's what I'm saying. And the question I had for you was because, well, the reason I reached out to Professor Ruby in the first place was because I was asking her if she had some like talks or anything that I could like reach, like getting more exposure to like gynecology generally. And then she offered me that I could maybe like work as a little help her a little side project. I mean, it's not a little side project, but in like so that it wouldn't interfere with my lab work. So, but she said to get your blessings, so I'm coming to ask her.
Sure. No problem. We
I
are
know,
two
but
friends and we like to collaborate.
yeah,
If we can collaborate with you, then
yeah,
We already collaborate. What are you talking
no, I mean
about?
to her.
Yeah, because that's the fortune.
oh yeah, I've been
Yeah, because we have this getting all the single cell it's not trivial. And because of beauty studies, you want to do one by one. No, so far the undergrad students clean up.
yeah, no, I texted Fortune because I also asked her about the other stuff. So I guess I'm just like asking to like tell me when there's like a meeting so that I can like see.
He's a team lead.
But I'm already in the group chat for some reason, so that's very nice.
Yes.
Yeah, and so thank you. Also, hope it's okay that I just do
Yeah, sure. We're more than happy.
so there's an expert talk.
That means, again, another promotion for NTU is that you have a very dense, you have a very denser lab, and also a machine learning lab, and to collaborate with us. So I think this is a really good.
My approach is like always open.
Definitely more expensive.
That was very
And second is that go for the money. If you can get scholarship. Yeah, that's
I still have time because all the applications are for this next year are like now, and I don't want to do it right away. So I still have like time to do it.
All right.
What you don't
I heard.
Okay, so let's come back. And Professor Ruby still want to share a little bit about 10 to 15 minutes about the computation method. So we'll let her share that, and after that we'll go to the in-depth Q&A and the panel discussion. Okay, sounds good? All right, so Ruby, please go ahead.
Yeah. Lingzi told me if I don't talk about computation, you guys will be like, man, what is she here for? Okay. Because I wanted to really set the ground, because this is not an easy concept for non-biologists. Even it's not an easy concept for biologists, right? Even so, I'm talking to a bunch of computation scientists. So I want to set the ground. I hope I explain myself clear enough, right? So after that, then, if we want to understand how the spectrum is built, how the plasticity is established, then one crucial point is which state is the cell at. You need to know where at this spectrum. So you would definitely need a quantification or any sort of scoring or any sort of phenotyping scheme, right? All right. So phenotyping, this is more qualitative, okay? Qualitative. So this is one way of doing it, phenotyping. This is what I did like a decade ago. So we took a bunch of ovarian cancer cell line, all right, 40 of them. And then we just took 3 prototype of markers. These are protein markers, all right? One is pan-cytokeratin. This is epithelial marker. And VIM, the vimentin, this is mesenchymal marker. E-cadherin, this is another epithelial marker, all right? So we utilize the staining. This is immunofluorescence staining. Okay, you stain the cell, and then you look at the cell and say whether they are stained positive, negative. So the first decision tree is the E-cadherin positivity. If it's a positive, because I said this is an epithelial marker, right? Then the cell is epithelial cell. If it's a negative, it's a mesenchymal cell. And then the second decision tree is, then we utilize this mesenchymal marker, vimentin. So if it's a vimentin negative, E-cadherin positive, then we define it as a true positive epithelial cell. And if it's an E-cadherin negative, vimentin, and then, back. So the first layer, decision tree is E-cadherin. So by E-cadherin, we decided whether it's if it's positive, it's epithelial. If it's negative, it's mesenchymal. And pan-cytokeratin is an epithelial marker. Vimentin is a mesenchymal marker. So if you are a true epithelial cell, truly an epithelial cell, what you would be having is pan-cytokeratin positive because it's an epithelial marker, but vimentin negative, right? Plus the E-cadherin positivity. If you are a true mesenchymal cell, then you should be negative for the pan-cytokeratin, which is the epithelial marker, and you will be positive for vimentin and negative for E-cadherin. So by 3 markers, you will actually have 4 phenotypes, right? So then you would have cells which are double positive for pan-cytokeratin and vimentin, double positive, meaning that they have both epithelial and mesenchymal markers. So they are in a hybrid state because they have both markers. but because of whether the cell has E cadherin positivity or not, you can roughly like categorize them into intermediate epithelial or intermediate mesenchymal. Is it clear? It's very clear, right? It's like you use a very simple 3 markers and a 2-layer decision tree to come up with 4 phenotypes. But this is very rough, right? You are categorizing. We're categorizing. Let's come back to the gender, binary spectrum. It's like 100% female, half female, male, and then male, 100% male. Sorry, your question?
Yeah, how do you name the different like beam and cat, like those are what are you like to categorize?
Those are okay. These are well characterized protein that are already known whether they express it.
Just the name in English.
The name.
Forgot.
Okay, so VIM is vimentin.
But those are used to categorize like the state.
Yes, those are used to categorize the state.
Binary results. Like it's only a binary result.
Yes, so you have the two layer of binary result.
My question was if you have 2 states that are intermediate E, you won't have any chance to like compare them together. It will be just the same state.
You mean the intermediate E? Do we compare?
If you have if you make 2 experiments,
Yes.
and at the end, like for the E cat it's positive.
Yes. Yeah. Yeah.
And then for the V it's positive.
Yeah.
You have in your 2 experiments 2 different states, and like so in the blue results,
Sorry.
you won't be able to like compare the 2 states together further.
I'm not sure I understand. You want to compare these two?
So it's like if you do the experiment for like 2 different cells,
Uh-huh.
and you want to know the states.
You just stain them. You stain the 3 together.
What?
You have 2 different cells, right? You want to know these 2 for each cell. So you stain the 3.
Yeah, and if you end up with the same result for the 2 cells.
If you have the same result for the 2 cells, they will be the same category.
Yeah, so you won't be able to know which one's, I don't know, more.
Yes.
Because it's a spectrum. It's not
Yes,
a binary.
yes.
My question was, is there some like further experiment to like distinguish those?
Perfect. This is my next step. Yes, so that's why this is phenotypes, right? The phenotype is giving you a very rough categorization. Here we only have 4, right? And then you're right. If 2 cells, they can be at the same state, like the pure E or intermediate E, but they might be different. You're absolutely right. And then, so, does it help to have more markers? It will help. Okay, because we only have 3 markers. Imagine you have more markers, you will be able to have more states, right? So this is another example. This is another study. They use the animal models in mice. They utilize proteins that are expressed at the cell membrane, and they use the flow cytometry, the antibody-based flow cytometry, to see which antibody binds to the cells. Here they have different combination. Here they would have EpCAM, this is epithelial state, and then vimentin, this is mesenchymal state, right? Keratin, this is epithelial state, and then you will have these cells that express positive epithelial EpCAM, and you will have this types of cell that are negative for everything. And then you will have cells start to express different mesenchymal markers. For this one, it start to express the CD106, and another one start to express CD51, and you will have another cell that co-express these two. You will have a cell that start to express a different one, CD61, and you will have another cells expressing 3 positivity. So it tells you that what markers you use to identify the phenotype would also affect how many categories you categorize them into. Simply, like previously, I used 3 markers. I have 4. Here there are like 1, 2, like several different markers. They come up with 1, 2, 3, 5, 4, 6, 7 different states. Again, are they the same within the same marker category for 2 different cells? Not necessarily, right? Okay. Another example, also phenotyping. This is to take the circulating tumor cells from the breast cancer patients. In this case, they use the RNA in situ hybridization. So this is not protein, this is RNA, using RNA. Using the transcript in situ hybridization, meaning that just to hybridize, see whether they express. And then they are very smart. They utilize a bunch of epithelial transcript and all gave them, all labeled with green fluorescence. And the mesenchymal transcript, they are all labeled as red fluorescence. So with this spectrum, it's very easy. You will have an E, pure E with no red, and by the intensity of the fluorescence, you can quantify them. Is E more than M, E equals to M equals to E, or pure M? For this, you can quantify. But still, because you lump all the E transcript into the same color, you're assuming every transcript are the same. But in fact, they're not. They're not the same, right? So imagine if you give every E transcript a different color. This is exactly what the single cell spatial is doing. Different barcode, different color. So you should see a lot more gradient there, right? This again is like meshing everything here. And to be able to detect that, your limitation is what? How good your fluorescence microscopy is in distinguishing whether it's really E more than M or E equals to M. How do you count that? So your detection is the limitation is at how good your resolution, your fluorescent microscopy, your signal is. You have questions? No, it's okay, right? So this is what we call phenotyping. You categorize them into them, and then these 3 examples tells you that using different markers, you will end up with different categorization. So a couple of years ago, we published 2014, we decided to do a scoring. So this is like a fine granularity of the scoring. This is RNA based, this is transcriptomic based, all right? So this is from the bulk gene expression, and then we can come up with a scoring from -1 to +1. The closer to -1 is more epithelial, the closer to +1 is mesenchymal. I'll tell you how we do it. So this is a cancer-specific EMT signature. This is published. You can get this paper and read it. This involves a 6-step of doing thing. The first step is compute the enrichment score. How do we compute the enrichment score? We gather all the published EMT signature out there, everything, okay? And then we come up with kind of like a gene set, okay? But all the published EMT signature, not necessarily that will work. Some are crappy, some are not. But then how do you determine what are the good ones? So we utilize the gene set, we compute an enrichment score, okay? And then you align your cell lines or your tumor, your samples with that enrichment score of the EM signatures. And then we lined it up, and then we took only the top 25% for training. Okay, if it's a cell line, we took it to top 25%. If it's a tumor sample, we took the top 100 from both ends. And then you do a binary regression. Because if you want to detect the intermediate, you still don't go from the extreme, right? Binary. And then you do the binary regression. And then we go through several rounds of like leave one out, 10 blocks, permutation for that, to clean up that binary regression. And then we utilize the SAMROC, the ROC curve for each gene to determine whether that gene performed well. So by using that, we could trim the entire gene set down to a small number of cancer-specific signature. We do it one cancer at a time. We curate the kind of a broad signature, but we apply on like ovarian, breast, lung, one cancer at a time. And then, so each cancer type will have its own unique cancer-specific signature by this, all right? And then the final annotation for the score is to do the 2KS test to assign the score for each sample for that, all right? And then this circus plot shows you that we did ovarian, breast, lung, colorectal, bladder, gastric. We got 6, and then you can overlap these 6 based on their epithelial gene, their mesenchymal genes. And the circus plot shows you that we have the weight. The inner layer is the weight, and the outer layer is the gene. You can see that we only took those that highly weighted from the ROC, the ROC and SAM fold change, and then we took the intersected genes, and then finally we come up with a generic EMT signature that works for tumor. We have 315 and cell line 218, and then when you overlap it, 118. So we use a very traditional bioinformatic binary regression and SAMROC way to generate this signature. I will stop here, and then this is how we utilize this score onto the tumor and then the cell line. You can see that it gives you this granularity that for example, let's look at the ovarian. You have an intrinsic gradient for different tumor of ovarian cancer, right? But you also see another gradient. What is that gradient? The gradient, you can see it is from colorectal all the way to glioblastoma. So the colorectal, over 1,820 tumors, you can see that the median score is the most epithelial one, right? And then for the glioblastoma, neuroblastoma, the last one, the green one, over 500 tumors, the score is the most mesenchymal. Why is that? Why is that? Actually, upon seeing this, we are very confident of our EMT score because this reflects back to what the development tells us. Neuroblastoma, the cell of origin, is actually not epithelial cell, is neuron, is neuron. Okay, and if you look at the second one, osteosarcoma. Osteosarcoma, the cell of origin is your bone. Your bone is mesenchymal cells. And then colorectal, and then the second one, gastric. This is your stomach and your colon, right? Your intestines. These are epithelial cells. And then, if you don't forget, I don't know whether you guys know about this. For us, human beings, we have 3 germ layers. All right, ectoderm, mesoderm, and endoderm. What constitutes endoderm is your digestive tract, your stomach cells, your colon, your intestine, these are endoderms. Your lung, respiratory, these are endoderms, all right? And the mesoderm, your bone, muscle, blood, these are mesoderms. So endoderm, mesoderm, these are exactly the epithelial and mesenchymal. This is when the trigeminal layer is formed. EMT is responsible to execute this lineage differentiation. So when we come back to this, the mesoderm origin cancer, osteosarcoma, gives us high EMT score, more mesenchymal score, compared to the colorectal, gastric. These are the endodermal derived cancer. They are more lower EMT score, they are more epithelial. We know that this score is very robust because not only it reflects the nature of the cancer, it reflects back to its cell of origin, to the embryonic origin. So I will stop here.
Okay, thank you.
So I have a couple of questions myself. So this EMT score, based on the understanding, my understanding is that it's sample-wise, right? Each sample is a patient?
Each sample can
be a cell line?
It is a cell line. The bottom one is a cell line. You can actually do a single cell. We've been trying to use it on the single cell. It can be a bulk RNA-seq. It can be a bulk microarray gene expression. It can be like a single cell spatial spot. As long as one data point, it's one dot.
Oh, okay. So it could be, but in this study, it's one sample is let's say one cell line or one patient.
For the top, that is the tumor, right? So that's one dot is one tumor. One tumor.
One tumor
One
tissue.
tumor. Yes. So the top one, we did it pan cancer. So I think it was 15,000. These are the TCGA pan cancer data sets, and then down there the cell line is one cell line, one annotated cell lines. Yeah, but you can use it for one single cell, one single spot, as long as there's one data point. And this is a catch because this is microarray, this whole transcriptome.
Yeah.
Sometimes when the cells are highly abundant, lowly abundant, that will affect the scoring as well. Yeah.
Okay. Can you talk a little bit more about how to compute the score? I'm still not very. Yeah. So here you say prediction of cancer sample and prediction of cell line. You say it's binary. So what exactly are we predicting here? So what kind of cell line it is? Is the classification?
No, epithelial state and mesenchymal state. We don't care what type of cell line.
Oh, okay. But how do you decide this state in the beginning, right?
So you need to know. You need to have prior knowledge of like this cell line should be epithelial state. So that is at the step 2. So you correlate with known EMT markers. We still need to have some benchmarking markers there to help us guide, okay, like with a high expression of, for example, ecacterin or what, this cell line here, these are the epithelial line. So you still need to have some reference.
Okay, so can I say from the machine learning jargon is that you first humanly annotated those data points, and then
Humanly annotated, yes and no, yeah.
You annotate the ground truths, right? So you have
Yes.
some ground truths
Yes.
of these samples, and you try to build a regression model,
Yes.
a logistic regression model to
Yes.
predict that. And in the future, when you talk about the score, you are just applying a new sample.
Yes.
And just apply this prediction model.
Yes.
It will produce a probability between 0 and 1.
Yes.
And that will be your EMT score.
Yes.
Oh, okay, I see.
there are several ways to the final score. We can still go back to this pool and then kind of utilize this as your reference, and then you use your the 2KS test to precisely give your new sample a score with the reference of this, right? Or you can utilize a probability, which is so this is absolute. This is from -1 to +1, meaning that we won't need to scale it because it needs to be, it will be scaled against the entire background reference of this, right? But let's say if you don't have this, you just have the signature and you have a new sample of your expression, you can still run it. But then it's a probability.
Yeah.
Yeah, then it's not an absolute -1 to +1 scale, but it's still workable.
Yeah, but we can scale it, right? We can
Yes.
scale it later.
Yes.
Okay, so but the key idea is that you need to first have a ground truth label and build a model to learn from that ground truth label. And for new samples, you just apply it and got the probability.
But this ground truth is a bit different from the phenotyping. The concept is similar, but this ground truth we kind of yes, we examine I think 6 or 9 transcripts that are documented, and then to pick those reliable cell lines as your ground truth for the binary regression. But
Okay.
the step of binary regression is just to clean up the gene sets, because when we curate the gene sets, you would have a lot of crappy genes out there being described, but are not robust enough to be utilized as a scoring.
So you're using that to do feature selection, essentially?
Yeah.
Selecting the feature, only keeping the genes that are relevant?
Yes.
Okay.
And because the end goal is to have a cancer-specific signature and also a generic one, which supposedly could be applied for every
all the other
all the
cases.
others.
Okay.
This actually not only for cancer. We now proved you can utilize it for normal cells as well, even though it's derived from cancer. So that's why, like I said, the entire spectrum for the development is important. It's a very important another ground truth, if you like.
Okay. All right, thank you. So let me save time for the students. So student, do you have a question? Yeah, let the student ask first.
Oh, can I just say, do I have to use the microphone?
No, it's okay.
Can you, thank you for the presentation. Can you give us like some specific examples how this can be applied to like the clinical studies or like in the practical? Oh, thank you. Yeah, so for example, you can talk like predict like more precisely predictive development of the cancer states or like the applied of the precision treatment.
One example is it correlates with clinical outcomes. So for the entire exercise, I didn't have time to go through it, we correlate whether high EMT score versus low EMT score, patient would live longer, or patient would have responses to chemotherapy. Now that you can imagine if you have a cohort, you have patient responsive to immunotherapy or what kind of therapy, you can utilize the score to look at it. So in this case, for that entire cohort, we have low EMT score showing better overall survival in pancreatic, gastric, glioblastoma, and ovarian cancer. So in fact, yes, we did have an assay being we developed an assay when I was in Singapore, FFP-based assay. We shrink the gene down to 105 transcript, and then we can build a scoring system from one patient sample to give a score. So aiming to predict the outcome. Other questions?
Yeah, so, okay, go ahead.
Yes.
But we can also use it for spot.
Yes.
So how reliable is this
VCM, multiple cell, you have one spot. Tang, you want to comment?
I think this question based on the concept of the VISM or whatever technology that we are working on. For example, for the VISM, is it a mixed cell? It's not the single cell, yes, but the signature or the gene expression from each spot is also depend on the proportion of the cell in the spot. So most likely we reflect the gene expression of the tumor because it's the tumor anyway, so it cancer cell is the most abundant signature there.
Sorry, I cannot find it. I have too many slides. But the question was, we can do it in a single spot, right? And the VCM, this is one platform of spatial proteomics. So it's like a spot, but each spot you will have a mixture of cells. So each spot is not a single cell. So you will have a mixture of different types of cell, and that is random because the technology is how you okay, spatial omics profiling. Here. VCM, yes, VCM, here. So this is a spot, this is a spatial omics, the spatial transcriptomic that Fortune is asking. So the idea is you have each spot, and then you have the cells, you have the tissue kind of like transferred to the spot. And then your hybridization of each spot corresponding to, it will just correspond to whatever cell happen to sit on top of that, right? So it's purely random because the random comes from how your technician transfer it to that position, right? So your coordinate for each spot, not necessarily even though it's the same tissue, you do it 2 times, not necessarily will be the same because it's very difficult to transfer to the exact same spot, right? So even for your same spot, same coordinate, for 2 slides, you might have a very diverse cell composition. And then for each spot, because the spot here, okay, so this is the most highly defined, high definition. This is my 2 micron spot. 2 micron is smaller than one single cell, right? But if you have an older version, the older version is like you can have 100 micron or 50 micron. 100 micron is like 3 cells, right? And then 50 micron, 2 cells, 2-3 cells, and then you can see the spot. They are space. So you will have cells that are outside the spot, and you will have, so because it's purely random, you will have a part, like half of the cell in the spot, half of the cell not be covered by the spot, right? And still you are computing for that. So for that, you assume that entire spot is homogeneous, but in fact it's not. So that's why he said you need to use the deconvolution, which is you can deconvolute using other algorithm to say, okay, within this spot, what are the percentage of epithelial cell versus non-epithelial cells to sort of infer that. Yeah, but so far from his computation, the correlation of the EMT score versus the composition of the epithelial and mesenchymal cell composition sort of correlate quite well. Yeah.
So you told like that you deconvoluted the spots. So like when you deconvoluted the spots, they are basically, then you extracted some gene information from that parts, right? It's how did you like deconvoluted the
You need to go back to basic markers. So we talk about the libraries, right? How do you deconvolute this, right? For each spot, for example, each spot you might have a mixture of epithelial cells versus immune cell versus fibroblast, right? Then how do you call them out, right? So you need to use the standardized fibroblast library, standardized T cell library, standardized epithelial cell library as your kind of your signature, right? And then you come back to that spot and to call the percent. It's kind of like you would have the kind of like enrichment of each signature within that whole library, that spot of library, right? And then from that you infer this might be 70% epithelial cell, 30% fibroblast, right? So it's again by the probability computation. So you infer that. So it's not absolute. Yeah. Does that understand?
Yeah.
Does that so you still need to go back to some benchmarking gold standard library that you rely on that marker, right? So in a way, it's not completely like you still need to rely on pre-existing prior knowledge for that.
Yeah, so to me it's kind of like learning from that, right? So you use that signature, use that library to first label. You still have to label them, right?
You still need to label
You
it.
still need to label its epithelial or mesenchymal.
It's like
right?
it's like your single cell. After your single cell, beautiful UMAP, right? Each cluster you need to say, okay, this cluster is what? Then how do you determine this is what? You need to, like, how do you describe a woman? Almost like that, right? You need to go to that ground truth.
Yeah, but to me this is a little bit chicken and egg because you don't like the binary thing, right? So that's why you need the score, but you're still using sort of the binary, but many binary, right? You have 10 markers, you have 10 binary, right? You're using that to give you a ground truth label, and then you're learning from that, and then you give a probability to say that, okay, we can now predict somewhere in between. But to me, it's still learning from that.
Yeah,
that multi-dimensional binary.
Yes,
right?
exactly.
Yeah.
Yeah.
So like when you are doing like CK scanning of the images and then you are like labeling it manually and then you are comparing it with the genomic library, like the manual labeling with the genomic library and like try to extract the gene information, right?
Say that again?
Like
we stain them manually?
like
No.
labeling them manually.
Oh, you mean like this, labeling?
Yeah.
Okay, we don't do this because you mean labeling each spot?
Yeah.
My pathologist will kill me. But if you read papers, we do have papers that actually they ask their pathologist to annotate each spot. It's very painful because he knows that each spot is 50 micron, and then you ask the pathologist, say, okay, give me the readout of this. You probably need to give the pathologist more than a bag of Labubu. Yeah, so I think, but they are now AI, there are Gen AI, the foundation models that can do it now.
No, there is nothing, I guess, because
No.
in my lab we are working on the same project. It's really
No, but
difficult.
I know they are like Faisal in Harvard, in Brigham, hasn't he got one already?
I don't think so because I didn't read about it yet.
Yeah, no, for Visum we never manually annotated. It's impossible. What you can do is like grossly, like, okay, this region looks like epithelial cell. This region looks like mesenchymal. That's it. And this region probably you have some immune cell. That's it. Each spot you will kill your pathologist. Yeah, but we do have other spatial project that we could annotate because those are it's a geomet, like it's like this, right? For this, you can annotate them. This is much easier because you select the region of interest is much bigger. So you can actually annotate like, okay, these are the epithelial cell, these are the immune cells, the rest are the rest. You can at least categorize them based on your marker expression, your different segment. This we can annotate. Yeah, but for the Visium spot, but I thought I read it somewhere. No, okay, we talked. Yeah.
Okay. Other questions from students? Yeah, Marta, go ahead.
So I have a question about the EMT algorithm, because it was developed initially for microarrays, right? For
Yeah.
microarray data. And then you mentioned that now you can do it for single cell or spatial omics. Did you modify at all the algorithm, or do you still use the same exact?
We didn't. We utilize it as it is. We utilize because the microarray. Yes, we didn't. But recently we find out that the quality for the single cell sequencing data would affect the scoring a lot, because the single cell RNA sequence, sometimes your coverage for the transcript is very, very low, right? And then so you need to clean up a lot on the original single cell data to be able to call it. Otherwise, we recently just run through one exercise, it just doesn't make sense at all. So all the scores were very low. So the library, the original quality of the sequencing is very important. But so far the spatial data is not bad. The high resolution spatial data, the Visium data, the single cell spatial data, especially because for some of the single cell spatial data, they don't have the whole transcriptome, only small sets of genes, right? 1,000, 5,000, 6,000. Not necessarily those genes will overlap with our signature. Even though it's partial overlap of our signature, as long as it's not too skewed, right? It still works.
Okay.
So that means that I know there's a room for further refinement for the signature, because the final one I said was like 300 something, right? Yeah, 315 if it's a tumor. But for ovarian, we can narrow down to 105 on an assay base. We could further narrow it down further, I'm sure. Now the smallest one you can run is how many? Not yet, okay.
I think Martha's question is also about the difference between microarray expression data and RNA sequencing. Although they are all measuring gene expressions, but they have very different unit, and maybe you need to do different normalization and things like that. So can you just directly apply?
What I can tell you is, we now have one experiment. This is on single cell multiome, is using the chromatin accessibility together with RNA from the same cell. From the same cell. So you have two readouts, the chromatin accessibility and then RNA. Okay, and we think that and this is from the chromatin accessibility. You can see the two main clusters, and then when you apply the EM score, the EMT score is computed from our EMT signature using the RNA readout from this, and you can see that this cluster, the right cluster, this is more epithelial. Is it? Yes. This is more epithelial compared to this. But you can see that it is still, this is too bright, but it's still heterogeneous there, right? So there are some noises there, right? So, so what we are doing now is that we have ATAC cluster, ATAC seq cluster, RNA seq cluster, so we have kind of like ATAC epithelial RNA epithelial, attack mesenchymal, RNA mesenchymal. So you will have this combination of matrix. So not every cell will have the concordance. So we again utilize the binary regression. So we pick those clusters that are AERE, AMRM, meaning they are concordant for both the ARTAC, chromatin, and RNA for the training. And then to come up with the new signature. Yeah.
Yeah, so to me, it's more like you're learning from the more confident. You first have biomarkers, right? You have those biomarkers, and so that you can separate them into different groups. And you're learning from the more confident one. Definitely positive, definitely negative. And then you use the learned model
and
to
the
assign
rest
the
yeah,
rest.
The rest is in between.
Okay.
Right. But one thing very interesting from this exercise, this ATAC and RNA exercise, is that, okay, we look at how this conversion rate, if we just assign the cell based on the RNA only versus the new ATAC plus RNA score, we want to see the conversion rate, whether they are still the same calling of the state, right? And we see that for the epithelial state, we increase the dynamic range. The epithelial, the original is about, for example, this set is from 0.41, then we can increase to 0.78. And the next one is from 0.52 to 0.67. But the mesenchymal, this dynamic range is the difference between the epithelial versus the mesenchymal, all right? So if you look at the minimum versus the maximum readout for the score, you can see that the max readout sort of like plateaued. Our original is 0.19, and then our new one, this one is 0.15. This one, the original one is 0.31 to 0.15. But if you look at the epithelial score, you see a much more significant change of the dynamic range. So why is that? It is not that your marker is not good. It is because the epithelial gene and mesenchymal gene, at the DNA level, at the genome level, the regulation of how the transcript are made are different. Because the epithelial genes rely on chromatin changes, chromatin accessibility, while the mesenchymal gene does not rely on chromatin accessibility change at all. The mesenchymal gene rely on the enhancer and promoter looping, the DNA looping, because the DNA is not a linear, it's a three-dimensional loop. It relies on the looping. So it tells us that if you want to predict anything by using the different mode, the different like omics method, like in this case is a choice of chromatin accessibility, the epigenetic. Not necessarily a random gene would just follow, would make sense, right? You need to know that whether this gene, in terms of its transcription, really rely on the epigenetic changes or chromatin accessibility changes. Otherwise, building the model on the mesenchymal gene, like we show here, it doesn't really matter, right? But you can always argue that then the RNA is good. But you can see that by adding the chromatin accessibility into the computation here, our epithelial dynamic range actually improves a lot. We can identify more granularity of that epithelial state, meaning that we can have more fine resolution in that.
Yeah, makes sense.
Makes sense, right?
Yeah. Okay, other questions from a student? Yes, yeah, Sophia.
So going back to the EMT score and relating these to a question we get very often when calculating risk scores, for instance, how would you use the score in the clinical practice? because I'm having trouble linking the academic work, which of course is very interesting for us to both you to understand cancer development. But then if you're a real doctor, so how would you use it in the clinical practice?
You know, the more I know about biology, the more hesitant I am to utilize any of this in my practice because I know too much about heterogeneity. Because every sample you get is a biased sample. It's a random selection from, okay, the surgeons, right? Okay, you take a biopsy. If the surgeon remove the entire tumor, okay, then it's non-biased. But when it comes to the hand of pathologist or any lab person, it's a random choice of where to punch that specimen, and then to send it to the lab, and then to get the sequencing read, and then to compute the score, and then to infer back to the patient for her or for his, like general. So to me, and then for this biopsy versus the next biopsy versus the third, you would have definitely, heterogeneity there, right? So that's why I think the most robust clinical assay so far is still the DNA base, because if it's a driver mutation, chances of that penetrance throughout different progression, it is much stronger. So you have, okay, I shouldn't say you still have heterogeneity, but it's still much more robust than transcript, because transcript really changes very dynamically, right? And then not to mention proteins. Protein change, we are not talking about PTM, the post-translational multiplication here for the proteins, right? But again, we utilize protein to stain 3+, 2+, for ER for her to determine which type of breast cancer she has, right? But you will be surprised if you do a second biopsy for the same tumor to do the staining again, you will get a completely different result. So I'm not that optimistic in terms of using this immediately to link to the clinical care right away. But that's why I think that makes the AI so exciting, is that because somehow we probably can swallow more gray zone, like unknown areas. The AI will probably fill the gaps of what we know or we think we know in there. I don't know.
I think at least you can also estimate the uncertainty that you have.
Yes,
over
the
your
uncertainty,
decision.
yes. Because for any lab test, you have to have certainty, because you need to have a cutoff to tell the clinician, okay, this is above the cutoff, so this is abnormal, you need to act, right? But AI probably can help to fill that uncertainty. But again, coming back, are we sure the uncertainty is the right one? That the right answer? I don't know. Unfortunately, I don't have the answer.
But summarizing this, then currently doctors don't use these tools in clinical practice, right? Because of the level of
omics. For the omics, for the spatial omics, no, not yet. But for the gene sequencing, yes.
Oh, okay.
Yes, because that is almost like yes or no, like you have the mutation or not. Even you have one clone having that mutation, you probably can still prescribe the drug for that, right? But it's just that for this complexity, for this fine complexity, like for example, I always ask, like, Prof, what's the difference between a cell having a mesenchymal score of 0.19 versus 0.15? I don't know actually, because there's no data to tell me that whether this 0.4 of difference really has an impact on the treatment outcome. Or should we just go back to the phenotype, like the 4 types of categorization? Is it enough to correlate with the outcome? If it's helpful enough, then why bother doing this much more expensive, more complicated scoring? Because for that 4 types, all I need to do is to stain 3 proteins. One is 100 100 NTD, 3 is 300 NTD, so it's like 10 USD, right? Trump will love it, right? You cut the cost, right? So coming back to whether there's utility. Utility question, I don't think this is the question that any scientist in this room can answer, because that is not your job. That is the policymaker, that is the practitioner, whether we think this one would go, even though it's a perfect assay, but if no insurance payer is going to reimburse it, the utility is still low. So it's way beyond what we're discussing here, whether you can accurately assign a score, predict responses, so on and so forth.
Thank you.
There's another one.
I had the same question actually, but we need to go further. So I understood that the last interpretation about the electrochemical score. You have just a sample and you have a very precise score. What you do with it? What can you say about it?
If it's with those tumors that we already shown to have correlation with the outcome, then we can say that, okay, a higher EMT score patient probably would live shorter. Yeah, lower EMT score patient. Yeah, but we test a lot of different types of tumors, right? Not all cancer has a correlation with EMT score. So some work, some don't. Yeah.
Okay, I think your questions? Okay. Yeah, but Ruby, do you have to go really soon?
I'm okay for 5 more minutes.
5 more minutes, okay, good.
Final question is that we're talking about the survival, but like when did you test the EMT score? After treatment or before treatment, or like, because you need to know the survival time if you do the follow-up. Yeah, and if you test the gene, if you test it after treatment, how do you know it's because of the treatment difference or something to affect the survival?
It comes back to how we built that training model. All those tumors were obtained pre-treatment. Those were pre-treatment samples. So you're seeing the correlation. You're seeing using the pre-treatment sample to predict the outcome.
Yeah.
After
the treatment is because of
we don't know. Yeah. So another good study should be done is to have compare the pre and post, or when the disease recur and then you biopsy again, and then you see whether the score will correlate. So to make the long story short, what we now know that when the disease recur, the score will usually go towards the mesenchymal state, meaning compared to the primary, the pre-treatment. When the disease is more resistant, when the disease is more relapsed, usually the EMT score will go up.
Go to the end of
the mesenchymal. Yeah.
But you can kind of assume that people follow the same protocol. So if they have the similar symptoms, they will be doing the similar treatment. Yeah. So yeah, that's also one way to understand.
The Oreo is the most popular here.
All right, I think Professor Ruby really has to go. So without further question, let's give her a big hand again. Thank you. Okay, so again, next week will be our workshop 3, and we'll let you know when will be the paper literature survey soon. Okay. All right. So see you next week.
对啊，大家，我就说要讲一个，
对啊，我就是
比较是 computation 背景，
对，可是我就想说前面那个方向，
那个还是要讲，还是要讲，但你后面就不行。
view 的那个 idea 是一个，就是大家 propose 出来一个 concept，
然后针对某一个区块。
Exactly， 你要怎么样去 target， 而且你要 push 它到哪？因为整个光谱那么长，你要 push 它到哪一个地方刚刚好？这件事情是这样。
然后 push 的方法是用一些
不过是对对对，你可以是用一些不过现在有一些做法是不同的 state 它会 express 不同的 service model。那现在有一个叫 ecological conscious， 就是说抗体它会是认在 service， 然后那个抗体是带另外一个药，这个药是可以直接有些那个感觉，我的那个 antibody 会认这一颗有追踪到对，然后那个药是直接去它对是有那个，那可是你看我刚刚不是说有一些是3个，虽然3个，有些是2个，所以那你就比如说你这么精准，你就只能杀掉有带那个人的细胞，那剩下那所以就对，那剩下的其实是可以变说，现在有一种叫做 spillover effect， 叫做它死了之后，它会释放出死亡的讯号。虽然我没有带那个 marker， 但是死亡讯号会影响到，然后我开始我就死了。这个叫做 off target 的这种 bystander 范围攻击。对，就是对，就是这样的。所以这个是一个那就是说，你可能就要很清楚说，哦，你大概就区分到要程度，大部分细胞变成然后你再加那个，
它等于把全身
但是你全身的正常的细胞你不行，
对啊，就是针对那个。
来，你可以哇，这个非常
下一个课是3个小时，
就是月经课嘛。我就走到那个。
对啊，他们各种在看
对，后来我就想说，如果讲这个，我知道你讲
对，大家会
对，他到19号，不是10号，19，有2周。
uploaded the slides from our talk, so they.
You're always playing.
They have one robotics one is separate from nah, that's robotics. And we It's too much. Getting home in lab that what do you think we need? I think honestly good enough. I can enjoy the exchange semester. There are like every course here is like a lot of
What else am I going to do?
那叫通灵。那叫通灵。
Like you start your thesis in the second year or like late in the final year?
We went to the But I thought like if you're like master grades that we have like I need to complete on the 25 goes to India, then I need to complete around 52 years.
Oh, it was also like the right like response. I like took a look at it and I was like, oh, this is after we went for the mistake. I was like, I would ask a question like this. Like, this is perfect. Oh, that's good. That's weird. That's really weird. I'm still gonna smooth that.
Or you are watching that something in China is going on like somewhere. I wanted to
Yeah, it's just that everybody sits together. Yeah, so let's say but everybody is
Yeah, like a special model that just predicts missing values. Yeah. But what you would not suggest it. I mean, for 850. So, but if you set it aside, then maybe you should follow it.
So if you read this paper, they use some method how to deal with they provide,
yeah, it would be better, I guess. It's already a fancy thing to use. It already looks good if we have okay, yeah, but I thought it was per website. Yes, but that is over 7. Oh yeah, so like fancy, like jewelry. Yeah, it's probably already going to be very Can you talk a little bit more about what he's
But do they need to do another workshop or just
Yeah, I think that's true. Just be here and keep, maybe the workshop for 1 hour.
some projects
Yeah. It was 5
Sorry, I already ate
Oh, and also he said that this annotation slide, including this
thinking about this,
like general
because I just want to let you know that it's hard to find
like if there's any mutation or which is the subject, it's much cheaper than every that's better.
Yeah, I mean, that focus on, it's difficult to find that so we were discussing about I think for your background, I think this slide so that will be the focus of the feel that
like taking so
think about it.
Alright,
okay.
That's another place. Do we just limit the data on these sites? This is another it's like restricted this. Yeah, they just like it's like already know the answer.
that's kind of true that PDF works on that.
no difference between DIC it's much more easier.
But if we don't go with this problem with the science after removing the sites, first of all, there's still some other case. The one, do you know it? Yeah, I saw this. The second question was like, I saw it with the
So I think the first step would be
what is about expert talk, because she's in charge of the expert talk, I think you get some extra bonus points. So send an email to the so she knows.
Okay, but also for the previous.
For the previous one, I think they said
Oh, okay.
so our
done in research for like between 1 to 2 points of
Yeah.
So I was correcting your homework, and I was not sure because there are some questions that are missing.
Yeah, I didn't manage to
I wanted to make sure they're actually missing, and because maybe you had them on your
I know that it's my I didn't recognize my time well enough. I had to finish doing once.
Oh, okay, because if you have them missing, I mean, if you just put the code and not like after the report, I can still do it. Just in case that
Next
happens.
we need to identify the transcription of that region, and then like just train the model based on the transcription and match, or otherwise we can use the expression like, because like if we are looking for like not a specific channel, like the suppose if we are having something A, D, G, A, G, we know this thing is responsible for hypertension.
That's from
We know it, but like
typing pride.
but what happens
on
in
your
the
yeah, it was
the
really
teen
nice.
channel,
I love all the positivity and there was like no hostility.
So
Which is like in Europe, even there it's not always it's so large as well.
and the person is
My
having
country is a little bit more dramatic, but here it's like, oh, just be happy.
Yeah, it's amazing.
Like,
Yeah.
only one gene is not responsible.
Oh yeah. But like I was honestly amazed by how large it is. I expected
the same sequence.
Despite the weather.
Yeah, I think it's because I've been here for a while, so
Oh, you used to that.
when you're shiny, maybe you
I mean, I've only been to so I just don't like big ones. Yeah. But like seeing all the time, actually, I was like,
I hope I can go to
Which we need to identify something like even we can use app like the thing I am talking about. If we are having like big sequence.
Yeah, I mean, we can identify those ones next to how like other calculations.
Same process, like the same thing we will consider for the next process.
So if we already know what that's then we can just collect them. But what's the next step? If we want to
We can use those create our existing data, figure out like what is the expression of the use those instruments to big set of samples using the because we need to in the laboratories, people people are like able to identify this specific things are responsible for hypertension. So we can use that particular gene as a baseline and use that gene to identify this set of gene information. If it is expressing more regularly, that gene expression.
是成龙演的吗？这是成龙演的吗？有日本版的。
Or maybe take some help from
吊带。
我记得他，对啊，他都是穿吊带出来的。
他有什么T恤跟吊带？
这个就是他的那个
腰配。我只有这个，我就说我就这个。
啊，可以了，而且还
对啊。
可爱。
不然也是有没有发现他的裤子上面。
很正常啊。对啊。因为是很久没看，就会没在看。
对啊。Like the first thing we will face is the annotation, like how we are going to annotate our because our data set is consist of gene information, some probe information as well, the initial and the ending. And there is some other data formats as well. We are not going to touch it, so we are going to only work with the genetic information.
It's suggested because otherwise it will be really biased towards the yeah, we cannot obviously try to do model with something. I think it's quite obvious hard to explain why the model is good at the end. Yeah. I suppose.
它可能需要一点缓冲的电。
Normally put it if you beat the basic. I think the basic
然后我们以前对，然后以前都去说习，对，然后现在没有去说习，是因为有个学长毕业，然后我们就没有。而且我都不知道毕业几年了，才真的去。对啊，因为我们每年都吃饭至少3-4次以上，像师姐老师生日，然后还有毕业学生毕业，然后还有像这个就没有理由就想要去这里。收30次以上，密度很高，学期结束了。对啊，我们都这样。对啊，老师说他好像没有喜欢，他说他试了他就不点了，他说上太多了，就是他又会下来继续调啊，就是也会啊，就是说不要严格他说。
I think like you can upload it the Google Drive that we shared with us.
I'm not really
就是感觉一直在玩。
because that is a single that is a single file and yeah, single CSV file and over 20 gigabytes. I need to unload the game from my computer. 我的 my USB has some issues. something like this. So it's just like the format of the file. So where the file is still file. So each of them also has this format. Okay, and you need to have a really special format to load the file over 8 gigabytes. So if a single file will over 8 gigabytes, then select the size.
So we need to read some research paper.
And after
and like if we ever find some particular gene site that is responsible for the hypertension, and so we can say that like, this person is having hypertension and like this is not having hypertension. And if someone is having hypertension, we can use that hypertension like gene, like the gene site like that is responsible for hypertension to identify the person is having hypertension in like having a hypertension severe mild or serious then classification. Because if the gene of gene so you kind of wrote it that way, right?
Yeah, when I saw the program and said
What is the format?
Yes. Because I have a and maybe also from Thailand only read the line so if we are trying to find missing value then just collect the columns of that calculated set so that there are many outlets.
See your data.
I need to
the file is like 10 like thousands and hundreds and thousands of people who use their device and where they use it. And it's very, very Pandas has the capability to read CSVs in chunks. So maybe use this to split it. Pandas library can read CSV in parts. So we could use this to produce a bunch of small CSVs to like distribute works and then send them back to everyone has like one USB stick and has to put them in the middle and then you combine them to the full data set.
用的来讲的话，可能只有用以人来看的话。
But the problem is like how we are going to process this.
也不是很久，它只是因为它是应该是说每一个患者或是住院。
Maybe we will ask the professor like.
但是我们现在变成，那你要做些的话，变成
Because we need to upload the data.
I think there is some comments there.
没有比人怎么办？哦，对对，我用那个就好了，我用那个就好了，我用这个就好了，谢谢谢谢。我们之前的资料会是这样子的，然后这个 A B， 然后它的譬如说进到这个里面，
Basically zip bombed yourself.
它会有一个时间点，对不对？然后所以就是这样。
Robert tries to like load it.
那你现在的资料会变成是，然后在它是
512
它会变成这样。
How much RAM?
你懂吗？它这个方类它是
The RAM is
对
16,
对
right?
对，没有，就是整个资料进去，它这个就是所以它到然后或者是没有。我的 TCGA 是那它就他直接给你时间，就是说多长。哦，就是那笔资料到底是多长？
I think so we need to decide a timeline first.
对。可是他后面就没有那个时间点。我没有资料，我是抽到时间档。它上面只有写数字，我什么的，那我就是从我的时间档要到你的时间档。哦，他所以他那个资料，他那个就你们的那个 raw data， 对，它是有的时候它是这笔资料里面它是有活着。然后
Access is best if you want to run a code. Yeah. I think so. Because the RAM in Asus, I think so yours is VOO,
那我就换一个方式啊，这个是1年度去看，那你可以去看1月份、5月、6月份。对对。那就没有办法了，后面因为你后面是也没有办法时间。
所以你会知道说今年更新完了它的。
对对对对对。它就是一个风水，它是一个风水，它是一个。哦，那你可能交换方式是上面这个方式，因为这个方式的话，它就是它只要有3个的话，它就会那你这个方式，对，拿走了，谢谢。
the next semester like one by
所以我一直在想，你们那个资料就可以变成这样，一样是变成这样子啊，就是你们是以月来看嘛，所以你这个人他可能就有这些，因为你到5个月他都是这个这个然后你逼这个人他，你们要切齐啊，做切齐是指说，如果你们高材生1个月的话，要大家都是5个月，但是他的资料他可能到第3个月，那第3个月他就是。
Okay, I uninstalled my games and went to the 100 gigabytes. So was the only public and so it's probably 50 or something. Oh, yeah.
如果你要进5个月，但你要3个月的话，它就会再发。对对。
我们就要去找到一个他存活是活着的，然后他的就是说他的 schedule，
Is the team winning in these?
Survival 很好。
就是说他家中的。
Do you play?
他说最后他我只能说可是他
的那个真正没死，指定5个月没死。
没事啊。
哦，对啊，那就是一样的意思啊，
就是他后面是每个月，但是他就说直接去
他这个就会升得掉，他就对，但是如果马上就5个月失去，就只能这样。因为你3个月是最多人的，因为你3个月，对啊，你只剩不念过，对，因为你现实的拿到
I try to play Valorant.
这样是蛮好的。
Very bad. I also played Overwatch, but I also suck at it. I only played tag, so I didn't have to. League of Legends I played do you know Heroes of the Storm? It's a game from Blizzard.
Are you going to meet your players tomorrow?
I'm going to meet them definitely.
今天会把分数投上去，你们可以看一下。其他的会写什么？你把它叫进来就好，可以过去你就把它叫进来。对啊，好吧，对，没错，答对了。没有啦，你要分析一些东西的话，你要看对啊，会比较方便。对，会比较方便。那你只要用，我们是本机的ES 号，没关系，下次可以采用ES 号，超好用，会打很好用。没有啊，ES 号更好用，而且它现在可以帮你绑，它可以连到那个电梯，然后帮你绑，然后你就可以让它摆说，可以，万家口键，对，已经有了，对，直接写成页可以啊，可是问题是他写的程式呢，还是要看，对，看是不是你要的，对啊，所以这样 OK， 好，试试看，对，你们还有一点点时间，
我看有跑一下那个我们那个 case 大概其实
那接下来怎么样？
No, but I like watching basketball. Really? Not NBA level. No, it's like everyone is telling me to see the what? Oh yeah, that's I heard that like the guy telling me like asking him how's the local baseball team here? Like beautiful girls. All right.
又在一体上面，然后你们的方法特别不同，然后他们的方法有什么小问题？
Basketball is popular. The actually is world championship owner basketball, yeah.
多有很多。这样就搜寻一下，然后你要搜寻开头的话，你要怎么搜寻？去哪 ？Google 烂死了，一个换一个，你去
Cricket match sometimes goes 5 days.
5 days.
Test match. It's like you start playing morning at 9 o'clock and it goes. Like the actual cricket is goes for 5 days, and then there is different formats. But the actual cricket starts in the morning 9 and then goes till 5. Now in Australia we are having like day night starts at 3 and then goes to 10 or 11. And then you go to sleep. Then next day again.
That sounds so bad. It's just like why? Just making sure, right?
So now we are having a short version of like 20 overs and like one day is the main.
哦，你是说，譬如说这些你觉得网页来去搜寻？我猜你们，因为你们有组织会，所以他应该是在 comment 里面搜。
In like the first 5 days is test cricket, that's when British start playing cricket. They started playing cricket for 5 days initially, and then they minimize it to 1 day, and then like
No transparent baseball.
Have you watched baseball? Yeah, I did, but cricket is very because in cricket your mental strength, like it requires a next level of mental strength. Because it's 5 days. Yeah, you need to stay there in the ground for 5 days. You can't go home.
So why don't we play chess?
Yeah, but the cricket is like even mental as well physical because you can't stay 5 days straight in the ground.
I still don't understand why you have to pay something for 5 days.
I don't know, but the concept of cricket is like the first inning, second inning, third inning, and fourth inning. So like every team, I think in baseball every team bats like for one time and then like 10 times I guess. One time and then another one time and then it's like, but in cricket it's 4 innings and that's very difficult to explain to someone. It's very hard game.
Okay, what's a couple of best format anything?
I think Germany also started playing cricket, but in the like the shortest format, like the few.
Of course, we have to work. But
哎，你们的目标要发文章呢。
television, you just like put on the television and just run in the background for all day.
Yeah, like initially people used to like listen to it in the radio.
Yeah, okay.
And then they started like telecasting it. So the 5 days cricket are mostly like in only in England and in Australia you can find people to watch cricket. But in India you won't see people are going there and watching cricket. Because to understand cricket, go to Australia or England. It's sometimes. But in India is the main like where people generate money from cricket. So people are billionaires in like, not like some people are billionaires and some are like they are paying really high.
Yeah, it's kind of hard, really hard. Who's the coach of cricket?
It's from India right now, it's Virat Kohli. No name. Yeah, because every country is but
the first, I would say cricket goat, the first is cricket goat of all time.
Yeah, it's whether say like the Sachin Tendulkar or the Virat Kohli.
Because he was a like hitter or is to the goat.
He is considered as the god of cricket. And he is I'm talking about this guy.
Yeah, okay. I think I've seen that guy.
And this is the legend, like he is a different level cricketer. Like his average in cricket was 99.96. That is unimaginable as a cricketer.
It's like the Michael Phelps.
Yeah, you can say that. He is from Australia. Like in cricket, like you score some runs. So every time when he comes to bat, he scores around 100 runs. And it's
400 runs. Wait, 100 runs.
Yeah.
So it's like he was. It's like in baseball. Like baseball, you basically hit the ball like 100 times in a row.
Not 100 times in a row. Like there is a 4 and 6 in cricket as well. Like you can run for 3, like maximum you can run for 3, like between the wickets. But there's
100 points. It's a perfect like sport.
Yeah, so in cricket we are having also 3 hours or 4 hours of
It's all mental.
It's too much stress if you think like one day you are feeling good, next day you are feeling bad, and everything is.
Can you swap out the players? I mean, the players have to like eat, go to the restaurant.
Yeah. They can like there in 5 days, even the like between 9 to 5 I am talking about. You will get rest like for 3 times, and every time for half an hour.
Imagine like a lunch break. Water break.
I think Australia and England are crazy. Like they are all like in every 2 years they play a 5-day
Watching for like an hour. I'm happy with it. I just like watching cricket. But it's crazy that you never picked up football. I mean, it's also a British sport, so
and because of the guy, like the god of cricket.
Okay.
How far did they come?
got it. It's harder than we got. Never. I mean, 15 years ago.
有的时候有一些时间会。